welcome everyone who's dialing in from Zoom or YouTube or wherever you come to us from. Today, I'm going to be presenting with Aaron Burton from Johnson Space Center. He's one of our special steering committee members who's here to help represent the meteoritic community. And he's also part of the OSIRIS REx mission as well. And with the landing of OSIRIS REx coming up next month, we thought this would be a neat opportunity to talk to those of us in the NfoLD community who might be wondering, what does sample return from meteorites and asteroids and things like that mean for us as a community? So we're going to just keep it kind of short and general, our presentations, because what we're really hoping to do is generate some conversation about how meteorite studies and extraterrestrial materials can be part of what we talk about in the life detection community. So let's see. The first thing I always like to do when I'm talking to life detection folks and I'm discussing meteorites is to try to emphasize the importance of meteorites and asteroids as representing primordial material. I love this comic from XKCD that really brings home the fact that all of the oldest parts of our planet that can tell us about what it was like here on Earth at the time of the origin of life is largely erased from the record. The oldest parts of our planet have been subsumed by the oceanic crust, and the oldest parts that we have are probably still on the moon. So that record of the origin in those conditions has been destroyed by these geologic processes. And that's what makes studying meteorites and asteroids special, is because they represent that reservoir. The other thing that I like to talk about is the fact that when we're talking about meteorites, we often wonder, where do they come from? And there are many different kinds of meteorites that we study here on Earth. Some of them are fragments of other planets, like Mars. There's lots of Martian meteorites. There's fragments of the moon. And then most of them, though, are from the asteroid belt. And I think Aaron's going to talk a little bit about meteorite taxonomy and some of those things a little bit more than I will. The other thing that we think about is the fact that some of these meteorites are from much farther out, and they might represent the rocky cores of comets. So this is a lot of the discussion that we hear from other sample return efforts, such as the Hayabusa missions, where that's a much more likely source of some of those other things that have been sampled through those efforts. Some of these might also represent failed planets. And they are, as I said, though, the closest thing we have to that primordial planet material. Of course, the big excitement right now is the OSIRIS REx mission, which is bringing back a fairly large amount of material from the asteroid Bennu. And what we're really wondering, there's lots of different efforts that are part of OSIRIS REx, but for us, we're really wondering what is out there and how does that relate to planetary material. So when we think about OSIRIS REx, some of the first things that many of us in the group wonder is, why go to an asteroid at all? If we have meteorites raining down on us, why do we need to go to an asteroid? But we're really trying to get back to that pristine-ish material that is an asteroid before it ever interacts with a planet or an atmosphere, like the kind of materials that are common that we can study here. So we're really getting back to those questions of, how did the solar system form, and what are the kind of materials that exist in the early solar system? And then, of course, how does life enter into that system, and when we're thinking of evolution in the early materials. Another thing to consider with OSIRIS REx is we're really wondering about interactions of large asteroids and our planet, and thinking about security concerns with asteroids. Like I said, there's lots of different asteroids where we have actually been. I think it's often easy to overlook in this era of in situ exploration, but we've actually done quite a lot of sample return, not even considering the moon, where we have kilograms of material down at Johnson, but there are many other solar system bodies that we have visited, and some of these here are things that we have brought back material from. As well as solar particles and cometary dust. So there's lots of great reasons to really try to perfect this idea of sample return. It means that we can know something about the pristine nature of our samples. We can know the geologic context. We can know much more about the material once we can use the kind of instrumentation we have in our laboratories here on Earth versus the kind of things that we can fly. This also represents a repository of material for generations of researchers and many, many new techniques to come. Thinking of the kind of efforts that have happened recently with the Apollo samples, where all these curated pristine materials were reopened up for people to propose for. So what is the mission? The mission of OSIRIS REx is really helping us get back to that early solar system and help us understand where we came from. And I realize now that some of what I'm saying is a little bit of a punchline for what Aaron will introduce, but I think maybe this can get us really thinking about what we might discuss as life detection scientists. I wanted to point out how far away Bennu is. It's pretty far from our planet. You can see it interacts with our orbit, but this gives you an idea. We're not clear out into the asteroid belt, but it still takes a couple of years for us to get there. Lots of facts about it. I think what's funny about this slide here is this is kind of an older slide. And if you've seen the video of the tag when the spacecraft actually collected in this kind of Roomba kind of thing, the material from the surface of OSIRIS REx, it really is a rubble pile. It's, there's not a lot of gravity and it's just barely held together. But that means that we were able to sample lots of material and lots of different sizes of clasts were collected into that tag SAM. So we'll have lots of different kinds of materials for us to interrogate for many years to come. The punchline for what does this offer to the life detection community is really getting to think about, with the kind of methods that we use, that we'll need to be able to back out all of that organic cosmic background that is replete throughout our solar system and doesn't necessarily represent life, but becomes something that we have to contend with in our measurements. And these kinds of materials and molecules are well represented in meteorites and asteroids. And really trying to get people thinking about how they might use the materials that are being returned by OSIRIS REx in the kind of studies they do. Because in 7 short months, in March of 2024, there will be a sample catalog that will come out that will have all sorts of information about all of the material that has been returned to our planet. The basic characterization data will be there. And you, as an astrobiologist, will be able to request and access these materials. So really be thinking about how meteoritic science can interact with whatever your discipline might be as well. And I'll let Aaron give us a lot more details on the kind of data we can get from meteorites.
All right, thank you, Heather. Let's see if I can get this to work again. All right, does that look okay? Okay. So for those that don't know me, I'm an astrobiologist, and for the last, I guess, about 13 years or so, I've been interested in studying extraterrestrial samples to help understand, how life could have started on Earth, or what chemicals might have been available for life to start here, and whether it could have existed elsewhere. So what does abiotic chemistry give us towards the origins of life? And so that's kind of a challenging thing. As Heather mentioned, a lot of the early Earth's history has been overwritten by plate tectonics and other processes. And so we're a little bit limited in, being able to see what the Earth was like 4.5 billion years ago. And so meteorites or various extraterrestrial materials like return samples give us a way to sample sort of actual prebiotic chemistry that happened in our solar system. And so on this figure, if we start with the diffuse interstellar medium, you get a cloud that comes together to form a star forming region. You get a star and planets that form. And then you get on one of those planets, you get exogenous delivery from things like comets, interplanetary dust particles, asteroids and planetesimals. Then you can also have endogenous synthesis, so hydrothermal vents, volcanic outgassing, Miller-Urey type experiments. So we could do a lot of those experiments as lab simulations to understand what the sort of endogenous synthesis might have been like. But it's really hard to constrain what the sort of extraterrestrial contribution could be. And so that's where the having these extraterrestrial samples can give us real authentic samples that we can look at in the lab. And then we do remote observations with various telescopes that allow us to observe chemistry that's happening, that we can't go sample. And so all these different approaches work together. But from a sample perspective, meteorites give us, most of our hands-on actual lab samples that you can analyze. So probably one of the most famous meteorites for origins of life and astrobiology kind of research is the Murchison meteorite, which fell in 1969 in Murchison, Australia. And so about 100 kilograms of mass were recovered. And on this chart, I've just shown all the various different classes of organic compounds that have been found in the Murchison meteorite, along with their relative abundance across that 100 kilograms. And so like aldehydes and ketones are present at about 30 parts per million total abundance. And then that number at the bottom, so that 3 grams is about how much would be contained if you dissolved all of the Murchison meteorite. So there'd be 3 grams of those. And so of interest to astrobiologists, we have these sugar derivatives. So a lot of them are sugar acids rather than the sugars we think of as like ribose and glucose, but they're the more oxidized form because those are more stable. We also have cyanide and ammonia, which are really good prebiotic synthesis reagents. Many of the nucleobases used in biology have been found. And then various cofactors. So shown here is pyridine carboxylic acid. But so that has been sort of cofactors like these. Nitrogen heterocycles have been observed. And then at the bottom left, we have carboxylic acids, which can make vesicles and different membranes. And then amino acids are what I have studied for a lot of my career. So the building blocks of proteins. These classes of compounds have all been found in the Murchison meteorite. And so one way to think of this for me anyway is like maybe these meteorites could be like a prebiotic bouillon cube that you can make sort of your prebiotic soup out of if it fell into the right body of water. So I'm going to talk about amino acids next for a little while. So I think one of the really interesting things to me, and I apologize, this is just a laundry list of chemical names, but more than 92 distinct amino acids have been positively identified in meteorites. And then many more have been detected, but they can't be unambiguously identified. So sometimes you get coding peaks, or there's so many possible isomers of a compound that you can't distinguish between individual ones of them. Among this, set of 92 that have been identified, and it's probably more now, 11 of the 20 universal proteinogenic amino acids have been found. So it's not a complete overlap, but there are many of the amino acids used in life that have been found. And there are many, many more that aren't used in life that are also found in meteorites. So another interesting sort of clue or topic in the study of meteorites is the idea of homochirality. And so in biological homochirality, all the proteins that are used in life, so that are translated by the ribosome, all use the so-called L configuration of amino acids, except glycine because it's not chiral. But it's been shown through the manufacture of synthetic proteins that you can make a protein with all D amino acids. And it will function in exactly the same way as a protein made of L amino acids, except if it accepts a chiral substrate, it will actually act on the other enantiomer, which is interesting. And so from this, it didn't have to be the case that you had to have all L amino acid proteins. You could have had all D amino acid proteins. It does seem that mixing the L and D amino acids doesn't work. And so that's an interesting observation. But one of the challenges that we've had is normally when you do like a Miller-Urey synthesis or other amino acid producing reaction, if you make chiral compounds, you tend to get a racemic mixture. So 50-50 mixture of L and D amino acids. And so how do we go from this sort of abiotic racemic state of amino acids to a homochiral state? Or more simply, how and why did we end up, or did life end up using only L amino acids? So the study of meteorites by many different groups, many different labs, many different samples has found a number of amino acids that are present in small enantiomeric excesses. And so shown on this slide are all the amino acids that have been detected along with the highest value reported of that amino acid excess. And so, there's L-aspartic acid at 60%, there's isoleucine at 50%. So some of these are pretty high. And those are always the challenge because if you see an L enantiomeric excess of amino acids in a meteorite, you assume that it's probably contamination because from biology, because that's the sort of the simplest explanation. And then getting to a more elaborate explanation, like there's some extraterrestrial mechanism for making these L excesses, you have to come up with a way to explain it and verify that it's not contamination. So I've shown L-isovaline here in the top in a red box. And so this is a chiral amino acid. It's not used in translated proteins. It does show up in some fungal peptides. But in those peptides, it's typically either racemic or only D is used in the peptides. And so it's been found in many meteorites in an enantiomeric excess up to 20%. And so here's a plot showing just sort of that range from as much as 20% enantiomeric excess to racemic, depending on the meteorite sample that you analyze. And this has been reported by both liquid chromatography, mass spectrometry, and gas chromatography and mass spectrometry by different groups, different labs, different samples. So this is pretty robust. If there is something going on that causes these isovalent L enantiomeric excesses, which is something that we're interested in because it suggests that there's some extraterrestrial mechanism for getting away from just the synthesis of racemic compounds. So for sugar acids, D excesses have been observed. And so this is taken from a paper by George Cooper and Rios from 2018. And so on the left are some GCMS chromatograms of, so these are sugar acids. So if you look at like lixonic acid and the top left panel A, you can see that there's a clear D excess. Xylonic acid is a clear D excess. And so these sugars aren't typically found in biology. They're not the like primary metabolic sugars, but it's interesting that they show up at D excesses. And then if you look at Murchison 39-2, so panel B, again, you see that D excess of, in this case, arabinonic acid, lixonic acid, and xylonic acid. And then if you look at the soil below it, particularly for the arabinonic acid, you can see that in the meteorite sample, there's a D excess, and in the soil sample, there's an L excess of arabinonic acid. And so again, that suggests that it's not due to terrestrial or microbial contamination, that there's some mechanism in this, the synthesis or preservation of these sugars or evolution of them over time within the Murchison meteorite parent body caused this excess to be found. And then on the right-hand side, there's a plot of the D to L enantiomer ratio. So racemic would be a constant line right at that 1. And so the 6 carbon is extrapolated. So, but for the 4 carbon to 5 carbon, that's the average D to L enantiomeric ratio of these sugars. So there's a clear D excess for both the 4 carbon sugars and the 5 sugar acids and the 5 carbon sugar acid in Murchison. And so just as in keeping with the amino acids, it looks like where there might be a mechanism to sort of break that symmetry and favor the production of one enantiomer over the other. It looks like somehow there's another, or the same mechanism may act on sugar acids as well. So again, trying to keep it short and not talk about everything. Those are two big things that I'm excited about being able to look at in these Bennu samples is, do we see enantiomeric excesses of amino acids? Do we see enantiomeric excesses of sugar acids? So we've done a lot with the meteorite samples, but most of the samples we get are from Antarctica. A lot of the recovered meteorites, so they've been on Earth for hundreds or thousands of years. Even fresh falls. So we looked at the Sutter's Mill meteorite, which was recovered within days of recovery, but it fell near Sutter's Mill, California. It rained. And so these samples can get contaminated fairly quickly when they're exposed to the environment. And so in contrast, when we have these return samples, we have the contamination environment of the OSIRIS REx touch and go sample acquisition mechanism head. We have that really well constrained. It's been well characterized. We have witness materials that have been flown along with the spacecraft. So if we find something interesting, we can look and see if that looks like contamination or if it's authentic to the sample. We also have archived test samples and materials from every stage of construction of the spacecraft. So we can go look again, at, well, what could possibly explain this? Let's make sure that if we do find something interesting, we can't come up with a plausible explanation due to contamination. And then another thing that's interesting, and Heather mentioned, the orbit of Bennu relative to Earth, and they overlap. So by necessity, all the meteorites that come to Earth have to have intersected with the Earth's orbit. That's that's how they get here. And so the meteorites that we get now are the ones that are currently falling on the Earth, and not necessarily the ones that were falling on Earth, 4.5 billion years ago during the origins of life. And so going to an asteroid purposely and understanding it, being able to characterize that, and then hopefully being able to look at other asteroids with telescopes and sort of back extrapolating, might give us a way to study more of the asteroid belt than what we're able to just from the samples that fortuitously fall from Earth. And as one final thought that occurred to me as we were going through this is that Murchison has really shaped a lot of the knowledge of meteorite chemistry because it's been so large. And for return samples, I'm really hopeful that Bennu will sort of be that Murchison because a fairly large amount should be returned, at least in comparison to most return samples. So hopefully it will sort of be the defining, kind of set of returned asteroid samples that, will really be available for study. And with that, I will stop sharing and hopefully we can have a good discussion and hopefully I spark some questions from the audience.